Good morning, dear student, good morning. We continue the course of normal physiology. The theme today, digestion physiology. Digestion physiology. Let's start from digestion in a mouse and in a stomach. Questions. Digestion is a main component of function system on maintenance of level or nutritious in an organism. Next, digestion types. Next, research methods of digestion tract functions. A. In experiment. B. In clinic. Next question, digestive tract innervation. Next, digestion in a mouse. Structure and properties of saliva. Swallowing. Next question. Digestion in a stomach, composition of gastric juice. And last question, phases of gastric secretion. So, first question, digestion is a main component of function system on maintenance of level of nutrition in an organism. Digestion is splitting. Proteins are split to amino acids. Fats to glycerin and fatty acids, carbohydrates to glucose and fructose. The digestive tract, DT, consists of a series of muscular cylinders which are covered by an epithelium. The mouth, throat, gilled stomach, thin and thick intestines, a rectum, and anus are located one after another. Endocrine glands, saliva glands, gastric glands, pancreas, bilious system of a liver, and crypt of a small and large intestine are attached to them. The backbone factor is a level of nutrition in an organism which has become below the norm. It is learned by receptors and they transfer information to structures of the central architectonics, medulla, hypothalamus, limbic system, cerebral cortex. The other two working bodies are formed here. The main performer is the digestive tract. Absorption of products. Digestive tracts. Absorption of products of food components. Split begins already in the stomach, here. The main absorption happens is in small intestine. As a result, the level of nutrition increases. The useful adaptive result is achieved. Receptors report about the achievement in structure of the central architectonics, medulla, hypothalamus, limbic system, cerebral cortex, and this function system stops the existence till the next moment of emergence of a backbone factor. Thus, digestion is the FS main component on maintenance of nutrition level in an organism. Digestion types. Digestion can be intracellular and extracellular. The person has intracellular digestion only in leukocytes. Extracellular digestion happens to be hand and parietal membrane, when digestion happens in all departments of a digestive tract. Parietal digestion is only in small intestine. On a source of digestive joys, digestive can be autolytic, symbiotic, and genuine. At autolytic digestion, Digestive enzymes arrive together with food. The region is breastfeeding. At symbiotic digestion, a source of digestive enzymes in symbionts, cohabines. They are bacteria. They live in a large intestine. At GNU and digestion are source of digestive enzymes in the macroorganism. So, next question. Research method of digestive tract function. In the experiment for studying of the DT function, the fistula technique is used. The fistula is an opening which is imposed on a stomach. Pancreatic ducts, bile duct, intestine loop, lumen, at the same time are brought to an abdominal cavity, and it gives the change to receive the corresponding secrets for research. And in clinic for research of the gastrointestinal tract function of a person, we use gastric Secretion method. It is fibrogastroduodenoscopy. The gastric Secretion is entered per os and researches the upper departments of ADT. A colonoscopy, the intestinal searches is entered per rectum and researches underlying departments of DT. Also, coprology is used, studying of structures of fecal mass, enzyme, blood analysis, methods of a computerized tomography. So, next question, digestive tract innervation. Gastrointestinal tract, gastroenteric nervous system. It includes bodies of neurons which lie in a whole digestive tubus and form full reflex arches. These reflex arches can work without CNS influence. We distinguish two big plexus, intermuscular Auerbach's plexus, plexus mesentericus. It is located between layers longitudinal and ring muscle and process along all gastrointestinal tract. The second one, submucous nervous plexus, Meissner's plexus, plexus submucosus. It lies in a submucous layer. Both plexus innervate a smooth blood vessels and cells of an epithelium of a digestive tube. Value of one nervous system of a gastrointestinal tract is visible on the example of illness of Hirschsprung congenital defect of both plexuses. Babies with this pathology are incapable of emptying an intestine independently. The gastrointestinal tract innervation is also carried out by means of a vegetative nervous system, a parasympathetic and sympathetic innervation, afferent nerves, and a visceral afferent innervation. Parasympathetic preganglionic fibers comes in structure nervus vagus from a medulla and as a part of pelvis, nerves from sacrum department of a spinal cord. The parasympathetic system sends fiber to exciting and inhibitory cells of an intermuscular nervus plexus. Preganglionic sympathetic fibers begin from cell inside horns in sternal lumbar department of a spinal cord Their axon innervate blood vessels and approach cells of nervus plexus, inhibiting their exciting neurons. Visceral afferent begin in a wall of a gastrointestinal tract and is a part of the nervus vagus, nervus splanchnica, and nervus pelvis. Go to a medulla, sympathetic ganglia, and spinal cord. So, next question, digestion in a mouse, structure and properties of saliva. Saliva contains 99.5% water and 0.5% solids. The solids consist of anorganic and organic substances, amylase, nuclease, phosphatase, mucin, lysozyme, potassium salts, sodium salts, calcium salts, magnesium salts, chlorides, hydrocarbonides, immunoglobulins G and M, urea, ammonia, creatinine, free amino acids, and mouse. The nutrition is split, moistened, and there is an initial stage of splitting of carbohydrates with the participation of saliva amylase. Saliva is formed in three larger innate glands: parotid gland, submandibular gland, sublingual gland, and in gland mucous cheeks, a palate and a pharynx. Saliva is formed in two stages. At first, lobes of sialents make the isotonic primary saliva similar to the structure of blood plasma. At the second stage, during passing across the removing canals of gland, is ionic structure changes. pH saliva change from subacidic to alkaliscent. Salivation is regulated by CNS. The central of salivation is in medulla. The salivation reflex under the influence of smell and taste of food is stimulated. Big salivary glands have a sympathetic and parasympathetic innervation. At increase of a tone of sympathetic nervus, it is allocated with a little viscous and rich organic substance of saliva. At increase of a tone of parasympathetic nerves, a lot of liquid saliva is emitted. Next question, swallowing. Chewing and initial phase of swallowing are carried out by skeletal musculature. The center of swallowing located in a medulla. The innervation is provided with the trigeminal, glossopharyngeal, and vagus nerves. The food lump is forced back by the tongue that starts a swallowing reflex through mechanoreceptor. The lump gets into a throat, the nasal cavity, and airways reflexively closed. Stretching of the top part of a palate starts a peristaltic reflex which promotes the foot down. Both end of a thallic have sphincters, upper and lower. The lower sphincters protect mucosa of a thalate from gastric juice. So, next question, digestion in a stomach, composition of gastric juice. The gastric juice is produced by stomach glands located in its mucosa. The main cells secrete pepsinogens. There are proteases, proteolytic. A-side cells secrete the hydrochloric acid and castle factor. Roles of the hydrochloric acid. It Activated pepsinogen turns it into pepsin. Next, possesses bactericidal action, and promotes opening of a pyloric sphincter. Castle's factor is a B12 vitamin carrier. Mucoid cells produced slime. Mucin. Next question, phases of gastric secretion. A small amount of a gastric juice is developed at rest. It is basal secretion. At meal there is a stimulated secretion. We allocated 3 phases there. 1 phase is complex reflex, cerebral phase. In this phase, the gastric juice is allocated at a sign and flower of food and at getting food into a mouth. It is appetite juice. Second phase, gastric phase of gastric secretion begins with the moment of getting of a food lump into a stomach. It includes nervous and humoral components. Nervous component. The food lump irritates the stomach mechanoreceptors. An impulse from them comes to a medulla to course of a vagus and to initial mural nervous plexuses. From their impulses go to a stomach. The terminations of these nerves allocate acetylcholine, which strengthens secretion of the hydrochloric acid and pepsinogen. Humoral component. At getting of a food lump into a stomach, a hormone gastrin is emitted. It contacts the receptor on the acid cell and strengthens the secretion of hydrochloric acid. Hormone gastron, on the contrary, slows down the secretion of hydrochloric acid. And phase number 3, intestinal phase of gastric secretion. It begins with the moment of the thymus getting into intestinal. This phase also includes nervous and humoral components. Thus, there is mainly inhibition of gastric secretion. Nervous components. The thymus-irritated mechanoreceptor of intestine and an impulse comes to a medulla to course of a vagus and intramural ganglion. From them, impulses come to a stomach to fondant glands. Humoral component. Getting of a thymus into a duodenum gets stimulates allocation of secretin and enterogastron, which slow down secretion of hydrochloric acid. Next. Будем продолжать. Будем. Next theme, digestion in intestines, role of pancreas and liver in digestion, motility of gastrointestinal tract, GIT, absorption in various departments of a GIT. Question, this big theme. Structure and properties of pancreatic juice, regulation of pancreatic secretion. Next, liver role in digestion. Next, digestion in nestis and ileum. Next, secretion of intestinal juice, its structure, properties, secretion, regulation. Next, digestion in a colon. Next question, motility of a stomach, evacuation of gastric contents in intestines. Next, motility of small and thick intestine. And last question, absorption in various departments of a digestive tract. So, let's start. Structure and properties of a pancreatic juice, regulation of pancreatic secretion. Pancreatic juice Pancreatic juice has alkaline reaction due to bicarbonatase, pH, pandase, hydrogenyl from 8 to 8.3. Enzymes are a part of pancreatic juice: proteolytic, trypsinogen, hematrypsinogen, elastase, procarboxypeptidase A and B. They are developed by gland in an inactive form and activated only in a duodenum gland by enzyme of enterokinase. Next, consists pancreatic juice: lipase, split phase; amylase, amylolytic enzyme, splits amylin and glycogen. Regulation of pancreatic secretion. Nervous regulation. Vagus strengthen secretion, sympathetic nerves block it. Humoral regulators: hamines, secretin and pancreozymin. Secretin and pancreozymin. Next question, liver role in digestion. Liver produced a released bile, components of bile secretin, hepatocytes, and epithelial cells come into bilious channels. Out of digestion process, bile gases in a gallbladder. In the course of digesting bile, sphincter is removed through odys in a duodenum. 1 liter of bile is allocated per a day. The bile includes bilious acid. The emulsified fats participate in absorption long chain of fatty acids. Also, cholesterol, bilirubin, bicarbonates are a part of the bile. pH of bile is equal to 7.8. Bilious acid from intestines are sucked up in blood through a portal vein, come back to a liver, and there they secrete into bile again. It is hepatoenteric circulation. Hepatoenteric circulation. Bile secretion regulation. Secretion by hepatocytes, parenchymal, depends on concentration of bilious acids in blood of a portal vein. And the ductal secretion is stimulated by hamine secretin. Digestion is a nascence and a limb. In a small intestine, there are 2 types of digestion: abdominal digestion and parietal digestion. Abdominal digestion happens in a GERD cavity with a participation of enzymes of pancreatic juice, intestinal juice, and bile. At the same time, polymers of food are split to oligomer. Parietal digestion happens with the participation of enzyme of intestinal juice on a brush border of a small intestine. At the same time, oligomers are split to monomers and monomers absorbed up. Parietal digestion is carried out on the apical surface of the enterocyte. Here, in its membrane, hydrolyses are built in which complete the hydrolysis of nutrients. The enterocyte is covered with microvilli. Therefore, the area of parietal digestion increases. In the membrane of microvilli, hydrolysis and glycolyzed. Between the microvilli and on their surface is a layer of glycocalyx. Glycocalyx provided sterility and selective permeability for the medium located above the membrane of the enterocyte. Between the filaments of the glycocalyx, there are hydrolyses from the intestinal and pancreatic juice. Here they complete the process of partial hydrolysis. This was started in the intestinal cavity. Above the glycocalyx, there is a layer of mucus produced by goblet cells and fragments of the slucen intestinal epithelium. In this layer, many enzymes of pancreatic juice, intestinal juice are sorbed. Thus, the transition from the cavity digestion to the parietal digestion is carried out gradually through two important layers in the function respect. Layer of mucous overlays and a layer of glycocalyx. Then there is actually a layer of parietal membrane digestion in which the final hydrolysis of nutrition takes place and their subsequent transport through the enterocyte into the blood or lymph. Next question, secretion of intestinal juice. Its structure, properties, secretion regulation. Components of intestinal juice are produced by Brunnerov and Liberkunov glands, enterocytes, epitheliocytes, and Baker cells. In a small intestine, there are final digestion and absorption of food. Enterokinase, peptidase, the lysine aminopeptidase, nuclease, and other split peptides to amino acid, amylase, lactase, sucrose, maltase, lipase are a part of intestinal juice. Cations and anions are also a part of intestinal juice. Intestinal juice pH is from 7.8 to 8.0. Regulation of intestinal secretion is carried out by local reflex with participation of a gastroenteric nervous system. A nervous system with participation of a CNS, vagus stimulate and sympathetic nerves block, and with participation of hormones, secretin, gastric inhibitory peptide. Next question, digestion in colon. The colon is the GAT output depot. There are absorption of water and electrolytes, deposition and evacuation of fecal mass. Symbiotic digestion happens with the participation of gastrointestinal microbial flora. At the same time, there is an initial stage of splitting of fat, synthesis of previtamins, group A and B, formation of ammonia from urea, fattive decomposite of proteins. Next question, motility of a stomach, evacuation of gastric contents in intestines. Motility of GIT is directed onto movement of food, chopping of mixing of food, deposition of food. Smooth muscle of a stomach generate action potential in response to food arrival into a stomach. Peristaltic valves extend on the direction to pyloric part of the stomach. The part of valves feed other amplify and close a pyloric sphincter. It promotes the best mixing of contents of a stomach. The muscle cells of the stomach are responsible for a motility of the stomach. The muscle cells of the stomach generation, exhalation, enteral nervous system, Meissner's and Auerbach plexuses, vegetative nervous, vagus trenchant, sympathetic nerves block a motility of a stomach. And humoral factor. Humoral factor, hormones gastrit, motilin, stimulate secretin, somatostatin, gastrointestinal hormone, and adrenaline inhibit a motility of a stomach. Evacuation of humus depends on gastric and intestinal factors. At the same time, gastric factors stimulate evacuation and intestinal ones slow it down. Gastric factors: stomach stretching, soft consistence of humus. Intestinal factors: duodenum stretching, Kao reaction of content of duodenum, availability of proteins, lipids, and products of their incomplete digestion in duodenum, hypotonic or hypertonic medium in duodenum, hamines, secretin, cholecystokinin, and irritation, inflammation, injuries of duodenum. Next question: motility of small and thick intestine. The peristaltis of small intestine can be divided into propulsive pushing and non-propulsive segmental movement. Non-propulsive peristalsis movement promotes the best mixing of humus and digestive juice. We distinguish non-propulsive segmental activity, tonic contraction, pendular movements, and fluctuation of fibers. At the end of the digestion process, one or two powerful waves of contraction longitudinal muscles move the remain of contents of small intestine to large intestine. It is propulsive peristalsis. Motility of small intestine is locally regulated with participation of gastroenteric nerve system. Sympathetic nerves block, parasympathetic nerves vagus stimulated, and with participation of hamines, gastrin, secretin, glucagon. Motility of colon carries out two functions: moving the fecal mass forward and storage the fecal mass. Normal antiperistaltic happens here, from anus. And last question: absorption in various departments of a digestion tract. In the mouth cavity, product of food digestion are not absorbed because they are not split completely. In a stomach, water, mineral salts, small amount of amino acids, weak solutions of alcohol are absorbed. The main absorption of product of splitting happens in a small intestine. After full digestion of the carbohydrates arriving with food, there are formed monosaccharide: glucose, fructose, galactose. Fructose is absorbed passively into the blood. Glucose absorbed in two stages: secondary active transport, symport with sodium ions in enterocyte, also from enterocyte in blood on the mechanism of light diffusion. The galactose is also transferred by secondary active transport. Proteins are split into amino acids D and tripeptide. Amino acids are absorbed by primary active transport by means of special protein carriers. D and tripeptide are also absorbed by secondary active transport, symport with hydrogen ions, by means of non-specific carrier transport various peptides protein. Fats are split into monoglycerosides and fatty acids. Short and medium chain length fatty acids absorbed passively in blood. Long chain fatty acids with bilious acid form micelles in gut clean. Micelles come into contact with a membrane of enterocyte. Bilious acid remain in a gut clean. In enterocyte, triglycerides are formed from fatty acid which with apoprotein form chylomicrons. Get to a lymph by an exocytosis. Fat soluble vitamins A, D, E, and K are similarly absorbed. Water-soluble vitamins are absorbed by active transport with participation of special carriers. So, dear student, the lecture is finished. Goodbye.